everybody. So today we're going to talk about Chapter 3, Tools of the Laboratory, and this is Part 2, the microscope, window on an invisible realm. And of course, in the laboratory, you'll be using the microscope a lot, so you'll become very familiar with it. We're going to talk about some different types of microscopy and then a little bit about staining. And as I've already mentioned, Chapter 3 is really the chapter that goes best with your laboratory experience. And please use this chapter to help support your learning in the laboratory, as well as what you're learning in the laboratory to support the material in this chapter. So in this particular section of the chapter, you're going to learn about converting different lengths within the metric system, describing the earliest microscopes, the three elements of good microscopy, differentiating between light and electron microscopy. Of course, in our laboratories, we only use light microscopes, and then compare different types of staining and provide examples of each. Again, much of this we will go through fairly quickly because, again, you're covering it in the laboratory itself. So looking at microbial dimensions, we can think of things as being macroscopic, looking in terms of meters and centimeters. And then you have to consider things that are microscopic, where things are in millimeters or micrometers or even nanometers. And so what are all those measurements? So I encourage you to look at the measuring thing in your bonus options in the in Connect. There is a section that will take you through these individually. So 1 millimeter, well, 1 centimeter, for me, 1 centimeter is about the thickness of my pinky or the width of my pinky. So that's kind of how I think of a centimeter. A millimeter is 1/10 of that. So a millimeter is pretty small. And if you have a ruler, you can kind of see that. And then when things get even smaller, microns or micrometers are then a tenth of a millimeter. So they're 10 times smaller even than a millimeter. And a millimeter is pretty small. And then in nanometers, again, tenfold difference in size there. So looking at different things that we know, you look at the reproductive structure on bread mold. You can see these through a microscope, or a louse, which again you don't really see very often with your eyeballs. They're pretty tiny, these things, and so that's about the limit of our sight with our eyes. And even then, we really don't see these very often if we're not looking carefully for them. As we look at light microscopes, things like protozoa are fairly large. Red blood cells then are also fairly large, and then you have yeast, and then bacteria about tenfold smaller than a typical yeast cell. And then continuing on, getting smaller, mycoplasmas are smaller than typical bacteria, and then viruses, we have large viruses like HIV, all the way down to smaller viruses like the polio virus, and then we can get down to substructures like a flagellum, a prion, which is just a protein, or DNA itself, and then even smaller things that we can't observe, even come close to observing in our class. In our class, we're gonna be cut off about here at the E. coli size. We can see things in this range in our class. In this range, you would need to have an electron microscope, and then you need special microscopes for this. It's pretty amazing what they're doing with microscopy now, where they're actually able to visualize atoms. I saw a paper just yesterday or a couple days ago where they were able to have a visualization of both the matter and antimatter, or the two things at the same that are occurring in the same time, like this quantum stuff, and it's just pretty amazing, way beyond the scope of our class and beyond scope of my particular understanding of that sort of thing, but I find it fascinating. So the dimensions again of bacteria, typically bacteria are going to be in the 1 to 10 micron type range is what we're going to be mostly looking at. So viruses tend to be very small, 20 to 80 nanometers. The smallest bacteria is about 200 nanometers. Uh, Mycoplasma, the largest, is 750 microns, which is really pretty big. Um, Yeasts are usually 3 to 4 microns, and protozoas 100 to 300 microns. So you can get kind of a sense of the size there, and the fact that not everything's the same size, and there's a range of size. So when we look through, in order to observe things in microbiology, as we said, things you have to look at through a microscope. We can we use the microscope for magnification, and magnification is usually going to use a glass sphere that is um, designed in order to magnify objects or make them appear um, visible when you wouldn't be able to see them without the light. And this is because of the way the uh, lens is curved, and the shape of the lens is designed for that purpose. Now, one of the things that will happen though when light hits glass is it will refract or bend. And you can see that in this image here. The light comes in, it hits the glass, that it's bending, and as it leaves, it looks like it's not in a straight line, whereas of course we know the light would be moving in a straight line. And so we use this term refraction to describe the bending of light as it passes through the medium. And the greater the difference between those two, the difference, we call that optical density between those things, the greater the refraction is going to be. So you can imagine in our microscope, we have you have your slide with the specimen on top. You have the light coming up. This is air. This is your glass for your slide, and the slide for your glass is probably about 1 millimeter thick. Then there's a little bit of air again, and then this is your microscope. This is your eyeball right here looking. Yeah, my drawing is not the best. But anyway, so light waves are really, really, really small. So even in the air, the light waves are coming through. It's a certain density. They hit the glass. That's a different optical density. That's going to cause some refraction, some bending of that light within the glass. And then when it comes back out in that air again, it's going to bend again. So as we'll see later, it's one reason we use something called oil, so that we have it going straight from the lens through the oil into our microscope lens, the objective lens, and then that reduces the amount of refraction that's going to happen in this space here. Because we want to reduce refraction, make it go away as much as we can, because that's going to obscure and change what we're seeing, and we want to actually see the image as it is. So the first microscopes had very few working parts and were very simple, and they looked extremely difficult to use. If you look back to chapter 1, you'll see Leeuwenhoek's microscope. I can't even imagine what that would have been like to use. This seems pretty difficult to me. Later ones use multiple lenses and things like that. Added a second magnifying lens, so you're actually looking at a virtual image. They use light to amplify this and illuminate the specimen, and then condensers and iris diaphragms in order to control the amount of light coming into the microscope. This is kind of similar to the scopes that you have in class. Actually, the scopes you have in lab are nicer than this image. BCCC has really nice microscopes for their students. But the parts you should really pay attention to, the ocular eyepiece, this often has a 10x magnification. And then the objective lenses, there usually are 4 of them that you move by turning the nose piece. And those will range from 4x, 10x, 40x, and 100x magnification. In microbiology, we mostly use the 100x magnification with oil. We call that oil immersion. The mechanical stage is just where you put the slide. Remember, when you're moving the slide around, you're only going to move the slide around using the stage adjustment knobs, because that way you can move the slide without damaging the components of the microscope. We have the fine and coarse focus knobs as well that will enable you to focus on the specimen. So again, these are things you'll be learning in the actual laboratory and actually using, but you should be aware of these different things. So what are the properties of an effective light microscope? We want magnification, resolution, and contrast. So for magnification, we have the ocular lens, the one where your eyeballs are looking, and then we have the objective lens, the one that's closest to the specimen. The ocular lens is usually set at 10x. So 10x meaning 10 times is the 10 times magnification. And then the objective lenses again will have either 4, 10, 40, or 100x magnification. So normally when we're looking at bacteria, we're using the 100x, which means that the total magnification when we're looking at bacteria and things through our microscopes, we're using a scope that's 1,000 times magnified. So the things that you're looking at with the oil are 1,000 times magnified, which is why we can observe them. We can kind of get a sense of how small they are. So because of the 2 lenses, and then there's like a mirror in here that it's gonna send the image up. This is what's forming a virtual image. And the objective lens closest to the image is what forms the real image. So when we're looking, we are actually seeing the virtual image. So this is showing us how the light is coming through. So the pathway of light in the 2 stages, if the light's coming up through the bottom, you've got your condenser and your iris diaphragm to control the amount of light that comes up. It's passing through the specimen that's sitting on the slide. Usually you've stained it in some way so you can see colors. You have the objective lens here. The light's coming through. It's going to hit a mirror, bounce back, hit another mirror, come through the ocular, and here's the virtual image that you're seeing. And here's the real image that's actually on the slide. And the reason I'm making a point about that is you'll notice when you're looking at things through the slide, through the microscope, and you think, oh, that that cell I want to look at is over on my right hand side, and so you move it where you think it's going to go to the right, and it goes the wrong opposite direction. And the reason is because you're looking at the virtual image. You're not looking at the real image. So you just have to remember that as you navigate and look at the different fields through the microscope. So the magnifying power, this is again how we calculate it. I kind of did that a moment ago. The 10x is because the ocular lens is always 10x, and then you times that's what the x is, by the magnification of the objective lens. And of course, generally speaking, in the laboratory, microbiology lab, we're mostly going to be using the 100x with oil immersion, which gives us 1,000x total magnification. So I've mentioned oil and oil immersion, and I mentioned refraction and said, oh, okay, this is why we use oil. So this is kind of a close-up view. So, and you'll see when you're in the laboratory using the microscope, the objective is going to be just very close, almost touching the slide. Very often students will say, "Oh, it's in focus," and I'll look and I can see the objective lens is like this far away from the stage. It's like you can't possibly be in focus because when you're at focus at 100x, the objective lens is almost touching the slide. You don't want it to touch the slide or be banging into the slide, but it's touching, it's so close that just this thin layer of oil is all that's separating it. And you can see here, here's the slide, here's the layer of oil, here's the objective lens. And this is how we use the oil to reduce, oil will help us to reduce refraction, or that bending of the light. It's going to end up distorting our image, and it increases resolution. What's resolution? Resolution is the ability to distinguish or separate two objects from each other. So with our basic old eyeballs, we can usually resolve things to about 0.2 um, uh, millimeters, which is not enough in order to be able to resolve microorganisms. So we need a microscope in order to do that. And the oil helps to increase that resolution. The resolving power is the minimum distance that things can be apart from each other and still be distinguished. So microscopes rely on having increased resolution, and we want to have the best resolving power as possible. Our microscopes are going to be using visible light to do that, but you can also use other sources. You can use ultraviolet light, you can use uh, lasers, you can use electrons, you can use a number of different things for that purpose. We use just visible light, so just a light bulb with all the different wavelengths of the light that come out of it in order to do our visualizations. So when I say visible light, this is what I'm talking about. All of the range of light waves from 380 nanometers to 750. You notice when you look at the visible light, what you can kind of see is a rainbow, and this is really when things get refracted in such a way, and things you can actually see rainbows in the sky. But there's other types of of waves too in the electromagnetic system, things that are not visible to our eyes, but nonetheless still exist, like ultraviolet, X-rays, gamma rays, have increasing energy and can be increasingly harmful, actually, as these wavelengths increase. And then we have also radio waves, microwaves, infrareds as well, that are lower energy waves as they go through. So here's the effects of wavelengths on resolution. So here is a specimen being observed at low resolution, and you can see these are the light particles kind of coming around the specimen, and it kind of looks like a blob, right? So you have kind of poor resolution here. But when we increase the resolution, notice we can see much better detail in the specimen, and we're able to actually see better detail as well as we've increased and reduced the wavelengths that we're looking at higher, uh, shorter wavelengths will increase our resolution here. So here's the appearance in a microscope. Here's the appearance of a small bacterial cell. Kind of looks like two that are kind of smushed together. The appearance in reality is they're two separate objects because we want to be able to see the reality. We don't want them to look like one thing. We want to be able to distinguish two objects here. We want to be able to distinguish as much as we can the parts that are inside a cell and see as much detail as we can. And part of why we, in order to see that better detail, increase our resolution, increase that ability to distinguish objects, is why we use oil. I've already kind of gone through this and mentioned it. You'll also be getting this in lecture, or sorry, in lab as well. So, you might think it's important, since you're hearing it in 2 different places by 2 different people. It's kind of a similar story. So we use just a little bit of oil, is enough, and that will help to reduce refraction, as I said, the bending of the light, and enable more of that light to get up into the objective lens and then into the ocular lens and ultimately into your eyeballs, so that you can see it. Versus um, if you don't have the oil, you have more air, light bending as the light comes from out of the slide and hits the air again. It's going to bend again, and that's going to reduce the amount of light that's going through, and your specimens will not have as good of resolution. So things will look blurrier. You won't be able to distinguish objects as well as when you're using the oil. So always in our class, you'll always want to be focused on using the oil. So the next part of that, this is contrast, and this is the ability, um, to have differences in coloration or density and things like that, in order to be able to tell, um, objects from one another. And the degree of this is referred to as the refractive index. So this also takes into account the degree of bending of the light, the refraction that occurs for the light, and the higher the refractive index, the greater the contrast. So you want the higher refractive index in this case that, get better contrast. So how do we do this? And what kinds of microscopes can we use? We have different types of light microscopes. In our class, we just use the bright field, and that's the most commonly used type of microscope. There's also dark field, phase contrast, and interference. We're going to briefly talk about each of those. And then instead of using just visible light, you can also use ultraviolet light for fluorescent microscopy, or you can use laser beams, which also is using laser beams will give you sort of you also are using fluorescence in that case as well. And then this new super resolution light microscopy is using 2 lasers in order to make these observations. And so these things allow us to see better detail of things because we're using a different component of light. So here's what the 3 of the types will look like. And this is again what we look at in our class. This is bright field. And this is where you're just using the light, bringing it through the slide. In this case, it's an unstained specimen. This is some kind of a protozoa, um, much bigger than bacteria. So you can see the details, the internal details of the organism here. This looks like a live, unstained specimen. Most of the time with bright field, though, and in our class, we usually use stained cells in order to see and increase the contrast between the cell and the background. Dark field, here what happens is you put a block on the condenser that stops the light actually from coming through. So the background becomes dark, and the light that does pass through, then you can see the insides of the specimen, the internal workings, a little bit better than you can with the bright field. Doesn't really allow for great detail of the workings, but it does give you a different way of looking at it. And then phase contrast is where you're using you use these different kinds of filters to control how the light is coming in through the specimen or through the slide. And this allows you to do some further differentiation of the components and further detail of the sample through the microscopy. But again, in our class, we pretty much just do bright field. Interference microscopy is using a differential interference contrast microscope, and this allows for visualization of live unstained specimens. As we can see this amoeba here in three-dimensional detail. Pardon me. And this is using prisms in order to help focus the light, and that will help add contrast to the image. Fluorescence microscopy, again, you're using ultraviolet light instead of just a regular white light. And very often you will label the cells then with some kind of a fluorochrome. So something that when the ultraviolet light hits it, it gets what we call excited. And that causes it to emit this bright color, the reds and the greens and the yellows here. And this can be used for diagnostics of particular types of infections. You can use fluorescent microscopy to look at like viruses in a cell and things like that, labeling that way. Confocal microscopy is also using laser beams in order to visualize. Okay, so confocal microscopy again is using a laser beam here. So it's kind of similar in a way to fluorescence microscopy, except for you're using a laser beam. And again, the samples, the specimens that you're using have been stained with fluorescent markers, with fluorescent stains, either using antibodies that are fluorescently tagged or just using the fluorochromes themselves to bind to the sample. And then what happens is the microscope and you use a computer, you take a series of images and stack them. So if you think about when you look at the microscope, we talk about depth of field, and you notice when you tweak the fine focus, things move in and out of focus. This kind of uses that where you start at one plane and you take an image, and then you goes a little deeper, takes another image, a little deeper, takes another image, and then the computer stacks all those images together to form this 3D or detailed image that you couldn't get just with standard microscopy or even with standard fluorescence microscopy. And so it allows you to see intricate details of cells and cell structures using confocal microscopy. It's also, they use confocal microscopy for things like looking at biofilms. It can be super useful for that. So you can see how the different types of cells within that biofilm are interacting with each other. And then even adding further to that and increasing the resolution even more is this super resolution light microscopy where they're using two laser beams and then again, fluorescently stained specimens, and you can see exquisite detail within the cell structure. So electron microscopy is another type of microscopy. Again, this is not one that we'll use in our laboratory. It was originally developed for studying non-biological materials. So people were using it really to look at metals and minerals and things like that, taking samples of rocks and stuff. But then in the 1930s, biologists started using it. And so this uses a beam of electrons, and we'll talk about two different types of electron microscopy. And this is what actually enabled using electron microscopy is what actually enabled scientists to figure out how the gram stain worked and how peptidoglycan, the thick peptidoglycan of the gram positive versus the thin peptidoglycan of the gram negative. This was actually figured out using electron microscopy. Now, if you think of the timeline there, the gram stain was developed in the 1880s, so it was 50 years it was in use before it was really shown definitively as to what it was, what the differences were between gram positives and gram negatives. So the science sometimes things work and we know them and we understand them, but we don't fully, we gain deeper appreciation when we have better and better technologies in order to understand things. So electron microscopy allows for extremely high magnification, between 5,000 and 1,000,000 times magnification. You can see the finest structures in cells. And there's two essential types of electron microscopy. The first is TEM, which is transmission electron microscopy. And here, the electrons are actually being transmitted or beaming through the specimen. And what you observe then is the darker or more dense spots then will stop the electrons from coming through, where things are dense in proteins or other things. And the less dense areas, of course, electrons will pass through more easily. So here you can actually see this is the COVID-19 with an electron electron microscope and electron transmitting electron micrograph. This is a picture of that. We only can see these images through computers, and usually they are artificially colored. Just recently, they came up with an actual naturally colored electron micrograph, but normally they have to be artificially colored. And notice when you look at a TEM, it's a two-dimensional image, but you can see the outer workings of the cells. You can see the surface proteins of this particular virus. Scanning electron microscopy, scanning, what happens there is the electrons come up, they hit the specimen, and they bounce off of it. They're not passing through. They're not passing through like we said with transmission, but instead they hit and they bounce off. And so as the beam of electrons bounces off, what you end up with is a three-dimensional image, and you can see exquisite detail on the surface. This is the surface of the specimens. And so again, it allows you to see contrasts of things, whereas TEM, and maybe this isn't the best image of it, you can often see internal details, like if it were a cell or something, really be able to see the mitochondria and the plasma membrane and the components of a cell. Whereas in scanning, you can see the outer parts of the cell in exquisite detail. So those are the different types of microscopy. So now let's talk about how do we prepare specimens for microscopy. We're going to focus on preparing specimens for the light microscope because that's what we use in our laboratory. And again, I'm going to go very quickly through this because you are spending a tremendous amount of time in lab doing these very things. So pay attention in lab and use this lecture along with the lab again to reinforce that learning. So how are we going to prepare the specimen? Well, do we want to look at a live specimen or do we want to look at a dead specimen? Most of the time in microbiology, we look at dead stuff. We're going to kill the bacteria so that we can, and stain it so that we can observe it. But sometimes you might want to look at live things. You might want to see, is the bacteria moving? Or you might want to look at a sample of pond water or something like that and look to see how things are interacting with each other. So occasionally you might want to look at living specimens, but most of the time in our laboratory, we're looking at what we call preserved specimens, and those are going to be dead or fixed or killed organisms. The aims of the observer are going to be observation, identification, and movement, and those things are going to determine whether you want to look at live or preserved. Because, of course, if it's a preserved specimen, it's not going to be moving. So if you're interested in motility, you're going to need to look at a living specimen. We also need to consider what type of microscopy is available for us. So if we want to look at living specimens, there's a couple different ways we can do it. One of the most common ways is called a wet mount. Wet mount is super simple. You have your slide, you put a drop of the specimen in there in some kind of liquid media, you drop a cover slip on it, and you look at it through the microscope. You're done. There's some risks to that. If it's an infectious agent, you're risking contamination and some other things. Hanging drop, what you do is you take a cover slip, you put the specimen on the cover slip, And then you put little dabs of Vaseline, little tiny, tiny dabs using like a toothpick on the corners of the cover slip. You use a special type of a slide called a depression slide, and you can see it has a little bit thicker than a normal slide, and it kind of has a hole, a pool cut into it, a divot cut into it. It's not all the way through, but it's um, like a opening in it. And then you lay the depression slide upside down on top of the hang of the drop, and then you flip it over. And when you flip it over, that causes the hanging drop to occur. There is a video in your textbook. Watch the video, watch the demonstration that you get in the laboratory. It'll make more sense. But then what happens here is the specimen then now is hanging inside that depression, and you can look at it through the microscope and see things moving and things like that in your sample. Now, because wet mounts and hanging drop mounts are live, you haven't done any manipulation of them, you can really get a true you can really see truly the size, shape, arrangement, color, if any is present, of the cells, as well as you can observe motility or movement by the cells. So that's what makes this a really useful tool if you really need to know precisely those kinds of information. But most of the time in our class, we're going to be doing what we call fixed stain slides, where we're going to prepare the slides, make a smear, and then you're going to stain the smear. So a smear is where you take a little bit of the sample, the bacteria, and you spread it onto the slide, usually in the size of about a dime or a nickel. You spread it in either a drop of water or a loopful if it's in a broth. You'll let it air dry. Then you heat fix it. You run it through the flame briefly. The importance of heat fixing is it kills the cells, it sticks the cells to the slide, and it preserves their components without too much distortion. The heat will distort them slightly, but not too bad. If you can't do heat fixing, you can also use alcohol or formalin for fixation. But again, that's going to cause a little bit of distortion of the slide. We usually use heat. We use stain to provide contrast because if you think about it, most cells are pretty much bags of water. And so they're not going to have any color. And we want to be able to see them, and it's going to be easier to see them if we make them have a color. So that helps us for things to kind of stand out a little bit better if we stain them. And just by putting the color on them, it makes it just easier to observe. There's two different types of dyes that we can use. We can use basic dyes, which are also called cationic dyes. These carry a positive charge and are attracted to acidic, negatively charged components on the bacterial cell wall. So typically the exterior of the cell, whether it's a gram-negative or gram-positive, is going to carry a slight negative charge. So a basic dye that carries a positive charge, opposites attract. It's going to be attracted to the cell and bind to the cell. The other type of dyes are acidic or anionic dyes. They carry a negative charge of the bacteria, negative charge of the dye. It's going to be repelled. So negative stains aren't actually going to stain the bacteria. They're going to stay outside of the cell and leave the bacteria unstained. And so we can look at some example what that looks like, and you'll be doing negative staining in the lab. Most of the time, though, we're going to do positive staining. Negative staining, you can see in this example here, you can see the bright spots, that's where the cells are, and the dark spots in the background is what's been stained by the negative stain. So the bacteria itself will appear colorless and clear because the background is what remains stained. Examples of this type of dye, these dyes are nigrosin and India ink. And we can use these to do a capsule stain, or sometimes it's also used in spore staining. We don't usually use negative staining for our spore staining. We tend to do positive staining for that. The more common type of staining is the positive stain, where the color is actually going to color the cell. So the cells will be red, or the cells will be purple, or the cells will be green, whatever color you've applied to them based on the dye that you've used. And the background will remain clear. So the glass slide will remain clear or white, as compared to the background in the negative stain. Some different examples of basic stains are crystal violet, methylene blue, saffronin, malachite green. Um, there's a whole bunch you can use. And there's a whole bunch of stains that are subtypes of that: simple stains, differential stains, capsule stains, etc. So again, most of the time you're gonna be doing positive stains, and this is what that looks like. And then you can also do negative stains as well. So types of stains, or the most basic one is called a simple stain. It's just what it sounds like. You make your smear, you put on some color. In this case, you've got crystal violet staining E. coli, or methylene blue staining Corynebacterium. Either way, you just put some color on, and whatever color you put on it, whether it's crystal violet, methylene blue, malachite green, whatever color it is, that's what color the bacteria is going to be. And it's useful to be able to tell sizes and shapes of the bacteria, but a lot of times we want to know a little bit more. So most of the time we'll use a differential stain. Generally, the most common one that'll get used is called a gram stain. And this is the one that's going to differentiate bacteria as being either gram positive or gram negative based on cell wall components or cell wall structures. And we'll learn more about that as we go through this talk and for the semester. Another stain that looks at cell walls is the acid-fast stain. And this is a type of stain that can just help us to identify a type of bacteria called Mycobacterium, which is important because Mycobacterium causes tuberculosis, and it also causes leprosy. I just saw a couple weeks ago that there's a big increase in leprosy cases in the United States. So Mycobacterium leprae is something that you may need to be starting to look for in the clinical lab. Endospore stains, again, that's another special stain that's used to look for bacterial endospores that are made by the bacteria Bacillus or Clostridium. Not everybody can make spores, but these guys can, and if they are present, they can be potential disease causers. And then the capsule stain, it allows us to look for capsules, which can help us to identify bacteria. The capsule is an outer outside of the cell. It's an extracellular matrix called a glycocalyx, and it's very often made of carbohydrates. And so we can stain the capsule to look for that. And that, again, will help us to identify bacteria, because some bacteria can make a capsule and some can't. And a capsule itself can be what we call a virulence factor, where it can increase the likelihood that that organism causes disease. So again, the gram stain. This stain was developed over a century ago, like 150 years or so ago, and it is still the granddaddy. It's still used every day in the laboratory. The basic stain that's often used to identify gram positives, stain purple, gram negatives stain pink, because gram positives are going to hold the dye that you put on first, which is crystal violet. They are going to survive the decolorization of alcohol, and then you come back and counter stain with a pink dye called saffronin. And again, you're going to learn more about this in lecture or in lab. Acid-fast stains, again, are used to identify Mycobacterium tuberculosis and Mycobacterium leprae that causes tuberculosis and leprosy, respectively. So here is a stain. The red cells are the acid fast, the bacteria here, and the blue cells are non-acid fast. There are different stainings that you can do for acid fast stain, different dye combinations that are used. So pay attention to that when you're in the laboratory. Make sure you know whatever dye you put on first. I think often in our class we use carbol fuchsia as the primary dye and methylene blue as the secondary dye. Um, so you would expect the acid fast to be kind of a bright red or fuchsia, like what we see here, these guys here, and then the blue would be non-acid fast. But there are other strategies for doing this stain, so just make sure you're aware that the colors can be different. But you need to know what the first color applied was, because the first color is the primary stain, and that will be what's positive for that particular stain that you're doing, in this case, acid fast. The next one is a spore stain, and again, the colors can be different depending on what stains you use. We tend to use malachite green as our primary. So in our lab, the spores would be stained green. Here, they used a red dye as the primary, so their spores are stained red. And the vegetative cells, which you can kind of see here, are stained blue. In our class, we tend to use safranin as the secondary stain in our spore stain, and so the vegetative cells would be red. So again, pay attention to the colors that you're using. The first color is the positive for that dye, and the second color will be the counter stain or the negative side for that. So in the spore stain, you're looking at the ability, or you're looking for cells that have been able to produce bacterial endospores, particularly Bacillus and Clostridium organisms, and and you're able to distinguish those from their vegetative or growing cells, which are the ones that are still growing and dividing, whereas the spores they've produced kind of like to save themselves. But again, not everybody can make spores. You'll need to know who can make spores and who can't. Bacillus and Clostridium make them, pretty much nobody else does. But the ability to make a spore does give it virulence. And virulence, again, the ability to cause disease. There's a number of different specialized stains that you can do. You can do capsule staining. I mentioned that as a potential virulence factor. Flagella. Flagella enables the bacteria to move, which also will increase its virulence. This is what a capsule stain looks like. I think you'll be doing one in the laboratory. Normally we do. So this is a combination of a negative stain and a simple stain. So the most simple way to do this is you start out, you do you do your two stains. And the simple stain is going to actually stain first stain, the positive stain is going to stain the actual cell structures, like this part right here. And then the negative staining is staining the background. And the positive stain is staining the actual cell. And this part here that remains unstained is the capsule. And so you can see the capsule around this example. This is Cryptococcus neoformans, but there's other organisms that make capsules as well. And you can see this clear zone around it is the capsule, whereas these long kind of strings inside, that's the organism itself. And so doing capsule staining can be important and useful for helping to identify different kinds of bacteria. Flagellar staining, again, it can also be used to help identify this organism. This is Proteus vulgaris, and all of these kind of stringy things coming off of the bacteria, those are the flagella. And you can use flagella staining to tell What is the arrangement of the flagella? Is the arrangement peritrichous, meaning it's all over the bacteria? Is it monotrichous? There's just one flagellum, like one little kind of tail. Is it amphitrichous on both ends of the cell? What is the arrangement of the flagella? And so that's where this particular type of staining can be useful, because different bacteria that have flagella will have a specific type of arrangement of their flagella that you can then help to narrow down the type of staining that you have. So this image I like because it really kind of just shows us a comparison of all the different types of staining that we can use in the laboratory using a light microscope. So the very simplest, a simple stain, we have our gram stain, we have a capsule stain. Um, this is another simple stain, acid-fast stain, a flagellar stain, and then a spore stain. So be aware, again, we can use different dyes in a simple stain. Um, a differential stain means you're using two dyes, two colors, two stains, and then we have the specialty stains as well. So here's your concept check for this chapter. What is the total magnification? You should be able to calculate that, multiplying the ocular with the objective um, magnification to get your total magnification. What's the importance of resolving power? Why does it matter to be able to tell two things apart? The differences between simple and differential stains, and the purpose of the gram stain. So that brings us to the end of this section, and I will see you soon. Let me know if you have any questions.